This is a production of Cornell University.
Great, thanks. Okay, we're going to lower the lights because some of the slides I think will show up better. Thank you, Klaus. And I'd like to thank Klaus for inviting me to talk with you today. This is my very first plant biology seminar, and I'm happy to have the opportunity to tell you about some of our work. Now, in the title, I include the word art, art and science, because I've found over the years that working in plant tissue culture and transformation, there's definitely an art to it. There is a feel with experience for what should be done next, and there's also a lot of creativity involved. But as I think about it, I think the same is true for other areas of research as well. Okay. So these are some points that I'm going to cover during my presentation. I'll give you some background on tissue culture and plant transformation. I thought giving you some of the basics would be really good before I launched into how we develop the methods and giving you examples of the projects. Lisa Earle, as Klaus mentioned, I did my graduate work in her lab, and before her retirement, she taught an entire class on tissue culture and transformation. So I'll have just a few slides to kind of put you on that level so you understand why we do what we do. I'll use these current projects as examples of how we develop those methods. Grape is the most recent one in our lab. We've been working with Anurag Agrawal at Cornell on Asclepias. And also, as Klaus mentioned, we're working with Setaria viridis, which is a C4 photosynthesis grass that we're developing as a model species. And then I'll end by telling you some of the next steps for those projects. So the way I see it, there are 4 components to development of these methods. There's the plant tissue culture and plant regeneration. Even before we go into transformation, we have to have an efficient method for plant regeneration. And these are some examples. So this is potato. So for potato, we start with plants that are grown in tissue culture in test tubes, which is referred to as in vitro. And we put those stem pieces on a medium that induces this growth called callus, which is an unorganized mass of cells, somewhat analogous to a tumor. And then we tweak that medium to give the cells the signal to produce plants. And these little potato plants are then removed from the callus and transferred to a rooting medium. This is tomato. Tomato, we start with cotyledon pieces from seedlings that are grown in culture. And these are all examples of plant regeneration after transformation. So you'll see that not every piece of plant material is regenerating shoots. And then for Setaria, we start with callus that is induced from mature seeds. And I'm going to tell you about that in more detail later. And this is what we use for the transformation. And again, that callus is put onto a regeneration medium to induce these plants to grow. Once we have a good system worked out for the plant tissue culture, then we move into the gene transfer methods. A critical part of this, and can sometimes be a stumbling block, is in the rooting and acclimation to greenhouse conditions. We, of course, work all this out before we even try gene transfer methods. And patience, lots of patience. It takes a long time to go from start to finish. And as you'll see when I talk about grape, we're sometimes working with very small plant material that we have to use a dissecting scope in order to do the work. So when we start with working out a new method, we'll first go to the literature to see if anything has been reported for that particular plant species. If we don't find anything, then we look to see what is the most closely related relative, see if there's anything reported for that. If we don't find anything, which happens sometimes, we then go based on experience and also a lot of trial and error to find out the best combination to use. So for plant material, we have a number of choices, but for monocots, what works best are immature inflorescences, mature seeds, and immature embryos. For dicots, you can pretty much try any one of these types of plant material. One part where we do spend quite a bit of time also is working out methods to clean up that material. Now that culture medium I showed you in the slides contains sugar because they need a carbon source since they're growing in laboratory conditions. And if we don't clean up that material, there are a lot of, the material will be overgrown by bacteria and fungi, and it will kill the material. So we use bleach, ethanol, sometimes antifungal, antibacterial compounds, but there's really a fine line sometimes between finding the right method that will clean up the contaminants, yet not kill the plant material. Some of these systems are easier than others, but for something like for the Asclepias, we spent a lot of time trying to find the best method for cleaning up the material. For culture medium components, not every medium contains all these components. It's just an example of what you can include in the medium. The major part of the medium are salt formulations, and those salts are like nitrogen, phosphorus, potassium, whatever is needed to support plant growth and culture. There are a number of standard salt formulations that we use. One common one is called Meshuggah Skog, or MS, but there are even modified versions of MS. So you can find, you can purchase an MS medium that doesn't contain any ammonium nitrate, because some plant species are sensitive to the ammonium nitrate. So there are a lot of parts to those salts that can also affect the plant material. One of the most critical parts of the medium are these plant growth regulators. And there are two categories. There are auxins and cytokinins. And these are an example of some auxins, and these are some cytokinins. So when we're working with these methods, it's not only the choice of an auxin or cytokinin that can make a difference. It's the combination, it's the concentration, it's the ratios. Sometimes we'll start out in an auxin, but then move to a cytokinin. So it really depends on the plant species that we're working with. For a carbohydrate source, sucrose is the most common, but we do work with some systems where maltose or glucose works better. Gelling agents, there are different types of agars and gel and gum. And sometimes it's really hard to convince people that gelling agent can really make a difference. I think some people think it's just not an important part of the medium until we give them a protocol and we say you have to use a gel and gum to start out with, even though it's embolded and it's red. And then they'll contact me and they'll say the method's not working. And I say, all right, let's start troubleshooting. And then I find out that they substituted a gelling agent that we didn't recommend. I was like, well, really, trust me, the gelling agent can make a difference. There are also amino acids and vitamins that are sometimes required. Activated charcoal kind of acts like an antioxidant, where some of the plant material will turn brown when we cut it and put it in culture. And the activated charcoal helps absorb that. It also absorbs inhibitors that might prevent plant regeneration. And I worked with geranium when I was at Sanford's, and when we would cut the pieces of geranium and put them in culture, they would just turn black. So we ended up looking at a number of different types of antioxidants and concentrations in order to prevent the phenolics from taking down the material. And there's also coconut water. Now I don't see this as often now in recent literature, but in the earlier days, coconut water was used, and you can still purchase coconut water through some of the companies. And it contains, a number of sugars, growth regulators, amino acids, and some undefined components that really can make a difference in getting plant regeneration or not getting it. Now, in developing a transformation method, where we spend the majority of our time is in the plant regeneration phase. We don't start plant transformation until we feel that the regeneration part is very efficient. And what I would recommend is that at least 30 to 40% of your plant material are regenerating shoots. And that's because when you do an agrobacterium infection, not every cell is infected. So if you have a system that's regenerating a lot of shoots, you have a greater chance of recovering transformants in the end. So that's why I would say if you have something that's less than 30%, don't even try it until you get that percentage up. So I'm just going to, all right. There are 3 different methods that you can use for transformation: agrobacterium, biolistics, which is the gene gun, and as Klaus mentioned, the gene gun was invented here at Cornell. And basically what happens is there are micron-sized particles, gold or tungsten, that are coated with DNA and blasted into the plant cells. There's also a DNA uptake into protoplast. If you have a leaf, you treat it with enzymes that takes away the cell wall. You're left with the protoplast. And then there are methods to increase that DNA uptake into the protoplast. In our lab, we use agrobacterium for all of our work. We find that it works perfectly fine. It's a soil-borne bacterium. About 30 years ago, a group of scientists decided that since agrobacterium is engineering plants in nature, it transfers genes into plant cells to produce the metabolites that it needs to growth. Can they take that bacterium from the soil and use it in the lab to transfer genes of interest? And it worked, and that was like the whole start of plant biotechnology. From there, the number of reports just increased dramatically. There are different strains that you need to consider when you're doing the transformation. You need to think of it in a plant pathogen interaction. There are antibiotics, depending on the strain that you're using, that eventually will eliminate that bacterium, because if we don't do that after a transformation, the agrobacterium overgrows the material and kills it. And there's also different phases of growth we need to look at and concentrations. So when we make an overnight culture of agrobacterium, we'll read the optical density. And in general, it's about a 0.6 to 0.7, but for some species we found we have to decrease that by 10 times. So we're using like 0.06 or 0.07 because it's just very sensitive to the agrobacterium. Now vectors, this can really make or break a plant transformation system. Those are the shuttles that were used in the agrobacterium to transfer the DNA into the cells. We use electroporation. There are different methods that you can use to introduce that plasmid DNA in that vector, but we find that electroporation works just fine for us. There are different selectable marker genes, and I'm going to go into more detail on this when I talk about vectors. And those are genes that confer resistance to antibiotic and also herbicides, and then we include those components, either an antibiotic or herbicide component, into our medium. And there are different types and concentrations, and you have to look at timing. And there's just a lot of parts of this that you need to look at for your particular plant species. When we're working at a new method, we'll use something called a reporter gene. We don't jump in to just start using our gene of interest, because sometimes genes of interest can have a negative effect on the regeneration of plants. It affects their development. So we start out with these reporter genes. There's one called GUS, which is beta-glucuronidase. And the way this works is that if you have this in your vector, you do your transformation, the plants you recover, you can take pieces of leaves, put them into an assay solution. If it turns blue, it's successful. If not, it's back to the drawing board. And there's also the green fluorescence protein, GFP. The nice thing about GFP is it's, as compared to GUS, when you put your material in GUS, it kills it. But with GFP, you can actually monitor the progress of that transformation by using a fluorescence scope. And we're starting to use more GFP construct vectors in our lab now. And then the other part of the agrobacterium is the length of time with it. When we're doing a transformation, say we have those pieces of potato stem pieces, they're just dipped for 5 minutes. It used to be in the older protocols, they would leave that material in for like half an hour. But that ends up having a negative effect. It stresses the plant material. So we find that a 5-minute dip is perfectly fine, and then it goes back onto the medium for 2 to 3 days, and that's when the agrobacterium attaches to the cells and does the gene transfer at that point. Most often it's in the dark. We do all of our co-cultivation in the dark, but there are some reports where they use light. And it's also the temperature during that period is also important. It's not surprising that around 19 to 22 degrees Celsius works best because, again, it is a soil bacterium. That's how it evolved. And we find that we've done comparisons of temperatures and found that that range works the best. Okay, so for vectors. These are some examples of vectors. And the pCAMB one is a more complicated diagram. It's showing the restriction enzymes that can be used for cloning. And then I have a more simplified vector down here called panic. This was developed for switchgrass or panicum. That's why they're called panic. And we've been using these panic vectors for Setaria. You can see that on this plasmid DNA, there's this segment here that I'll discuss a little bit more in the next slide. And then on that backbone, there are also a bacterial resistant gene for kanamycin and also this origin of replication. This origin of replication can also be very important depending on what plant species you're working on, and we found that to be true for Setaria. This is a closer-up view of a vector, and what you see here is there is a left and a right border, and that's the sequence that the agrobacterium recognizes. Agrobacterium has endonucleases, and those are the sequences it recognizes to cut that DNA at those points and transfer it into the plant cells. Those vectors also contain promoters. I'm just using these as an example. There's all sorts of different types of vectors that you can use. This is the NOS promoter driving the NPT2 gene. Now the NPT2 gene is a selectable marker gene, and it confers resistance to kanamycin, the antibiotic. So when we do our transformation, we'll put that kanamycin into the medium. And the reason we do that is, so say we're doing a tomato transformation and we have cotyledon pieces. Just about every cell in that cotyledon has the potential to go on and to make plants. And with the selectable marker gene, only the cells that receive that NPT2 gene go on to make plants because we have kanamycin in the medium. if we didn't do that, we would have a lot of plants to screen in order to find those few that are really transformed. And it can also affect the efficiency of the system. So we do need to include a selectable marker gene. And again, it could be for antibiotic resistance or herbicide resistance. This is another promoter that is a common one called 35S. You could put your gene of interest here. I've put GUS there because that's what we use for developing methods. We then electroporate that into agrobacterium. We grow an overnight culture, and we do a transformation experiment. So these are some tools of the trade for doing that. There's overnight cultures. We do the electroporation. We plate it onto the medium. We use toothpicks to transfer those colonies into the liquid medium. This is a freshly inoculated culture. It goes onto a shaker overnight, and then hopefully you get growth. And then we take this solution, centrifuge it down, we get a pellet, and we wash it with a plant tissue culture medium because the salts that are used for growing the bacteria are much too concentrated and can have a negative effect on the plant material. So we wash that pellet with a plant-based culture medium. The next, as I mentioned, we'll work out the rooting and acclimation to the greenhouse even before we do transformation. We certainly don't want to be working this out with our precious transgenic lines. We prefer that we get the rooting in culture before we transfer it to soil. We sometimes need to use a lower salt formulation. Auxins, something like tomato, we don't need to use any auxin in the rooting medium because it roots readily. And also there are some systems where we use activated charcoal because if we don't see rooting, there might be an inhibitor there that's causing it to not root, so we'll use activated charcoal. For the acclimation to the greenhouse, there are a number of factors to consider, not surprising. Soil type, lighting, different plant species that we work with, we have to use different types of soil. And you'll see that these are some Setaria plants that are covered with water bottles. And the reason that they're covered is that when we take plants out of culture, they're in test tubes or they're in magenta boxes, which are a culture vessel that we use. And it's like 100% humidity. And the cuticle isn't well developed. The stomates are wide open. And if we transfer those to soil and we don't cover them, they'll wilt and die. So we had been using plastic bags in the beginning, but then someone had the idea that people in the lab are drinking water, we're having bottled water and Gatorade. So we thought, let's try those. And we cut the bottoms off the bottles. And we cover the plants. After a few days, we'll simply remove the cap. And then after a few days more, we'll take off the bottle. And then we wash those bottles and reuse them. So it's much better than plastic bags because it was difficult to reuse the plastic bags. We had to cut holes in the bags to gradually acclimate them and then discard those bags. So now we have a good supply of bottles in the lab. So this is a listing of the plants that we have worked with and those that are current. Those listed in the black text are those which are current projects. Those in red text are ones that we're doing transformation as a service. So in my group, I have support from BTI for two and a half people to do transformations as a service. And the way this works is if someone is interested, say in tomato, like Jim, we do a lot of work for Jim, they'll give us the construct. And we'll do the transformations into tomato, and then we'll give them the plants back for their work. And it's a fee recovery system, because we have to repay that loan back to BTI for what the money that they give us to support this service component. But then the other projects in the lab are funded through different funding agencies. And these in blue text are ones that we have worked on in the past, but they're no longer active projects. So I thought you might be wondering, how long does the whole process take from when I do the infection until I can get plants back? And when we do our transformations as a service, people are really anxious to get the plants back. And we do our best to be able to, give them the plants, and then they want to know how long it takes. So from the time, say, for tomato, that we do an infection of the cotyledon pieces, it takes about 6 weeks to go from the infection phase to having these little shoots regenerating. Then we transfer those shoots to a rooting medium. And if you're interested in fruit, it takes about a total time of 5 to 6 months to recover red ripe fruit. If you're interested in just looking at the plants for disease or insect resistance, obviously it's a much shorter time. For potato, it's roughly the same time frame. This month difference here is because some potatoes take longer in the greenhouse to produce tubers. But again, we start with infecting plant potato stem pieces. And then in about 8 weeks, we have these little potato plants that we can cut and root, and then we'll put them into the greenhouse and produce tubers in pots. But again, if you're just interested in the plants, it's a much shorter time frame. So that's it for your instruction on plant tissue culture and transformation. And now I'm going to jump into the 3 projects to use as an example of how we develop those methods. So as I mentioned, Grape is one of the most recent projects that we have in the lab. And I wanted to work on Grape since we started the transformation service, especially knowing that Grape is such an important crop in New York State, and that there's work going on in Geneva, and I thought we could really help those projects along. So this started as a collaboration with Gan in Geneva, and he suggested that we work with 2 model grape plants. One is called Pixie. It's a dwarf vine, and these were developed to help accelerate the grape breeding programs. Instead of having to go out to the field and working with vines in the field, these lines can be grown in the greenhouse. Pixie stays small. It's a sport of Pinot Noir. And the reason it's short, it's because it's a mutant, GAI mutant, and it affects the gibberellin pathway. So these plants stay small. The internodes are very close. They're really dark green leaves. Scout is an experimental line that was released by the USDA and ARS in 2010. It flowers continuously in the field and greenhouse. So you can see how this would be an advantage for research programs. You have a continuous supply of flowers. It was developed by a cross between Ruby Red and an Asian species, and this is the makeup here of the Scout. And then Genwan also recommended that we start working with Concord because it's an important one in New York State, and it's often used to make jellies and jams. And if you're interested in reading more about Pixie and Scout, there is a publication that was put out by Cornell that describes the background on that plant material. So for Grape, there are 2 possible sources based in the literature for starting plant cell cultures. The one is immature inflorescence, and that is what works best for wine grapes and some others. This method for the leaves from plants in vitro does not work for the grapes that are used for winemaking, but it's mostly seedless grape types that work through this method. But again, not all of them. And I'll show you why there are advantages to using the leaf system as compared to the immature inflorescence system. So we've been doing our work based on reports by Gray and Deckney. We start by inducing callus on the plant material, and then we look for something called somatic embryos. There are 2 ways where plant regeneration can happen. There's a somatic embryogenesis and an organogenesis. I'll show you examples of organogenesis later, but for Grape, it's through the somatic embryogenesis mode. And when we started this, we ended up testing 14 different media formulations based on what we found in the literature to find something to work for Scout and Concord. We're not doing Pixie yet, but for Scout and Concord. We had to look at different salts, different growth regulators, and other media components. Once we have that callus and we have the somatic embryos, or SE, are transferred to a maintenance medium. And this maintenance medium contains a high level of sucrose. Sucrose generally is around 20 to 30 grams per liter in a culture medium, but for this medium we needed a higher level at 60. There are no growth regulators in the medium, and as I mentioned before, your gelling agent can make or break a system. In talking with Dennis Gray, he said it's very important to use this agar called TC agar throughout the whole system to get a good response for Grape. Okay, now here's the fun part. So, this is an immature inflorescence from Scout, and this is a dime we use as a size reference. So you can imagine with that immature inflorescence, the size of that. And what we end up doing with these is we treat the inflorescence with a bleach solution to clean it up. And there's a lot of dissecting scope work that has to be done. Because what we need to do is take a bud from this inflorescence, cut it open, and then take out the inside flower parts. And what is most important in this system is that the filaments stay attached to the anther, because that is where you get the right type of callus, the callus that's going to give you that embryogenesis. It's difficult to keep the filament with the anther. What we found is by taking out the entire part here and putting that on the medium, we were much more successful at keeping the anther with that filament. We put it on a callus induction medium The callus forms at the connective tissue. And then that callus, we're using a dissecting scope, and we're looking for these tiny little white embryos that pop up. These tiny embryos are then moved to that maintenance, and it's actually a proliferation medium. And you can see all these little embryos, these somatic embryos that have come up. This in here, these little ones, are what we call a pre-embryo, and these are the somatic embryos. And it's these somatic embryos that are used for the transformation. I don't know, I'm really fascinated in a kind of a nerdy way by embryos, by somatic embryos. I think they're really cute, they're very white, and it's fun to work with the systems where we're getting somatic embryos. So as I mentioned, there's a lot of work at the scope, and this is Kate Pigeon. Kate spends a lot of her time, taking out the flower parts, looking for the somatic embryos. And I asked her to look at the camera to show that she can still be smiling after all this scope work. But in talking with Dennis Gray, he said that when he gives his protocol to people and they then say we're having problems, he finds out that they're not using a scope. They're just trying to do it without using a dissecting scope, and it's an important part of the process. So this is a close-up of some of the somatic embryos that are on that proliferation maintenance medium. And you can see these are more, if you lose the term, mature embryos. And down here, and this is a close-up, these are some of the pre-embryos. So what we do to keep the embryogenic system going is, again, we're using the scope. We'll select out these tiny little embryos, put them back onto the medium. And this isn't a black background. This is actually the charcoal in the medium, the activated charcoal. And then we'll take these embryos for transformation and then leave these as the source for getting more embryos for our work. So we haven't done a transformation yet, but we will be doing one in about 3 weeks. We needed to be able to bulk up those somatic embryos until we had enough to do a transformation. And according to people I've talked with and through the literature, what we're going to do is we're going to take 1 gram of the scout embryos, the somatic embryos, the more well-developed ones. We'll take 1 gram of those. They go into a liquid medium for 2 weeks on a shaker, and then that solution is poured through a sieve. The smaller embryos will go back into a liquid, and then we'll pick off those larger embryos and use this for transformation. We're going to use an agrobacterium strain called AGL1. AGL1 is what's called a hypervirulent agrobacterium. There are other virulent agrobacterium out there that you can use, that you can get a transformation. But what we found is with some of those other virulent agrobacterium that they're really difficult to eliminate from culture. But we find that with AGL1, we get a good transformation efficiency. We don't have a problem with eliminating the bacterium, and it also results in a lot of plants that are single copy. So it's important to not have multiple copies in your transgenic lines of your introduced genes. In AGL1, we find that we get a really high number of single copies, sometimes 2 copies of the introduced gene in our plant material. For the vector, we're going to use the NPT2 selectable marker gene. We use GFP because we really want to monitor the progress. And Dennis Gray told me that within a week, if it worked, we should be able to see the green fluorescence protein expression. And then we're going to hope for the best and see what happens before we can move on. So I mentioned that another method is for doing in vitro leaves from leaves from in vitro plants. And we decided to use this for Concord. And the reason we decided to use that for Concord is because getting a source of immature inflorescence was difficult. They're only at the right stage 1 time per year. So someone in Ganwan's group would go out to the field to get those inflorescences for us. Ganwan lives in Ithaca. He would drive to Ithaca at the end of the day. I would meet him out front of BTI. He would hand off a box of inflorescences, and we had that one shot at getting, trying all this different media to get it to work. So I started looking in the literature to see what else can be used. And I found that leaves can possibly use, as I mentioned before. It's more successful for seedless grapes, but we're going to try it for Concord. And the way this works is we take a young shoot from Concord plants, which actually we have now growing in the greenhouse. And then more dissecting work. We go in here and take out the small apical meristem. That apical meristem goes onto a medium. And when this plant is bigger, we'll go ahead and we'll take those leaves, we'll put them on a callus induction medium and see if we can get embryos that way. This way, we can have more of a supply of material to bulk up the somatic embryos that we need for our transformations. Okay, that's great. That's where we're at, and I'll tell you some of the next steps later. We're working on Asclepias with Anurag at Cornell. And the purpose is to develop Anurag is interested, and he thinks the community would be interested in having milkweed as a model system, and transformation is an important part of that, being a model system. He identified 7 different species based on their chemical composition and their insect interactions. So we started with these 7 different species. We really wanted to be able to use in vitro grown plants so they would be clean. We wouldn't have to worry about cleaning up leaf or stem material every time we wanted to do a transformation, but we couldn't get them to root. The milkweed just would not root in culture. And it used to kill me when I'd be on a walk and I'd see a milkweed growing in a space between the sidewalk and a wall. I was like, are you kidding me? You're going perfectly fine there and we can't get you to root in culture. So I could give a whole seminar on what we tried for rooting, but we gave up and we said, all right, we're going to have to use plant material grown in a growth chamber or a greenhouse for our starting material. Cleaning it up was a challenge. It was a challenge finding that's kind of sweet spot where it cleans up the contamination but yet doesn't kill the plant material. There's nothing more disconcerting as coming in and seeing a fungus growing all over your plates. Okay. So these are some images of the species that we're working with. This is the common one that you might be familiar with that the monarch butterfly goes through its life cycle on. This is A tuberosa or butterfly weed. And I think they're beautiful. Here's some other images. I've actually started a milkweed garden at home. These are really pretty. I really like the Kurosavica. But this is how we get to see them on a daily basis. These are some of the stem pieces that are making callus on this callus induction medium. We use the scopes to scan that callus to look for the starts of plant regeneration to make sure we're on the right track with everything that we're trying. So when we see something like this, we get really excited because this is the start of the production of shoots. Now, unlike grape, which is somatic embryogenesis, this is organogenesis. And when we see this, we know that we're getting to that point where we could recover plants. Although sometimes we're fooled and they're actually roots, and that's pretty disappointing. But this was pretty exciting when after trying all sorts of different methods, that we came up with these little shoots. So because working with 7 different species was difficult, and plant tissue culture and regeneration, there's a definite genetic component to it. Species, even closely related species, they require different conditions and culture. So we decided to just focus on the 3 now that gave us the plant regeneration first. A key part of the medium was using an antioxidant. We used DTT. We used agrobacterium strain, LBA 4404. This is showing some GUS response in the cultures, and we've done some recent real gene transformations where we're looking at knocking down COI1 expression, and COI1 is involved in defense. And right now, Amy, who works on the project, is doing characterization to see how far down that COI1 is knocked down. And then we'll continue doing transformations with the other species. The last project I'm going to talk about is the Setaria viridis project. This started out as a collaboration with Tom Britnell when he was at BTI. And as I mentioned, the interest is because it's a model species. It has a small stature compared to something like maize, sorghum, or sugarcane, and it's a C4 photosynthesis. This is a phylogeny of the grasses. Setaria viridis is here. It's a wild relative of the cultivated foxtail millet, and it's a close relative of switchgrass. It has a lot of attributes that make it attractive for a model species. It's a small stature. You can grow a lot of plants, kind of like the Arabidopsis or the dicot world. We can grow a lot of plants in a small area. And what really we needed at this point was to have a transformation method worked out. And Tom asked me if I was interested, and I said, definitely, we'll take on that challenge. So what I did is I went to the literature first to see if there was anything reported for viridis, and there wasn't, but there were reports for its cultivated relative Italica, and they started with mature seeds. The only problem with viridis is there's a dormancy issue if the seeds are younger than 3 months old. So we found that we had to break the dormancy. We tried all sorts of methods to break the dormancy, and what worked out the best was removing the seed coats, and I'll show you in the next couple of slides how we do that. The seeds are then cultured on a callus induction medium. We then clean up the coleoptiles and the roots, and we transfer that to fresh medium. And as I mentioned before, the importance of gelling agent. When we started out, in the Italica reports, they used agar in the medium. And what we were getting with viridis was this really sticky, gelatinous, non-regenerable callus. And what we really wanted was this more defined callus. And I knew from my prior experience with monocots, that sometimes a gel and gum, in this case phytogel, actually works better. And when we switched to that phytogel, we recovered more of this more well-defined regenerable callus. You couldn't ask for anything better for plant regeneration. Just about every piece of callus regenerates multiple shoots. We took one shoot and put it on rooting medium. It rooted well, it tillered a lot, and it grew just fine. So we knew that we were now ready to do plant transformation. So as I mentioned, to remove the seed coats, we came up with this really simple, but it works. We took a tray, we have a rubber stopper, we got some ridged plastic material out of my kitchen drawers, and we put the seeds and we rub that rubber stopper over the seeds. We were concerned that maybe it would break a lot of the seeds and wouldn't get good germination, but we got close to 100% germination. And then this is where more patience comes in. Those seeds are selected away from those that have the seed coats, and this is what we use for the callus induction medium. For the transformation, we use this vector. This vector has a gene for hygromycin resistance. NPT2 and kanamycin don't work that well for monocots as compared to dicots. We infect the callus with the agrobacterium. They then go to a selective regeneration medium that has hygromycin in it. This is a very happy day in the lab when this is our non-transformed control. We put it in GUS, no blue, but when we put one of the putative transformants into GUS solution, it turned blue. And this is showing some leaf material from a control, did not turn blue, and these are some leaves from some older transgenic lines. Now, as I mentioned, we use that selectable marker gene, because if we didn't, this is what we would end up with after we did a transformation. So we would have to analyze a lot of plants in order to find these precious few that are really transformed. We wanted to see if we could get this to persist into the next generation. So we let some of those transgenic lines go on to make seed, grew them in the greenhouse, harvested that next generation, which is called the T1, assayed them for GUS using the assay solution. And you can see again, non-transformed control and GUS expression in those progeny. So this is a timeline for how long the whole process takes. From culturing the seed on callus medium to having seeds harvested from your transgenic lines takes about 21 weeks. But if we had that callus already, we could cut off 6 weeks off that process. And with monocots, it's really difficult to maintain callus long term and still have it maintain that high regenerability. And when you do get regenerants, you sometimes see a large percentage of albinos because the transposons are activated. But we decided to try it with Setaria, and so far what we've seen is we're just testing taking callus, keeping it long term, and putting it on regeneration medium. And it seems to be acting differently than maize or rice callus, in that we're not seeing that hit on plant regeneration. And now we're going to try some of that longer-term callus for transformation. Okay, so I'm not going to talk about. I don't have time to talk about our CRISPR work in Setaria, but we have tried genome editing by targeting this flowering time gene called indeterminate 1. And what you can see here is that these are the controls, empty vector, no target in them. And these are 2 lines that were recovered. You can see there's a definite difference in the flowering time. So that was a really good indication phenotypically that we hit the right gene and that it's working. And all the lines now, both the primary ones and progeny, are being analyzed to see what types of mutations we had and the frequency on those. So it's been a lot of fun working with the CRISPR-Cas9 systems. So in summary, I hope I've impressed upon you all the work that it takes. Sometimes people don't appreciate it, and we do a transformation service and like, what's taking so long? How come we don't have our plants? I mean, that's rare, but there really is a lot of work that goes into the system. I have a great group. They're very patient, they're very creative, and that's why we are successful in what we do. So for the next steps, for Grape, we are going to do what I mentioned, the transformation with GFP. As soon as we have enough embryos from Concord, we're going to try transformation of Concord. For the Asclepias, we'll characterize those COI1 knockdown lines, and we're trying to recover more of those so we can have different levels of expression and different transgenics. Improve the rooting in soil. We've given up on in vitro rooting, and we're looking at rooting in soil. It's not great, but it's better than what it used to be. And we're also starting to work on the other species of the 7 species. For Setaria viridis, we're actually looking at other accessions besides the A10.1, because I mentioned that there's a genetic component to plant regeneration and also for transformation, and that we feel that if we can find another accession where we can get higher than 5 to 20% transformation efficiency, that would make it more high throughput for us. And as we're looking at the long-term callus maintenance so that we can cut off about 6 weeks off the process. And we're going to expand our genome editing efforts. So right now we're just using it for mutagenesis to knock out genes, but we're looking at some of the new CRISPR technologies that have come along to activate endogenous genes and do some gene replacement in those systems. And we're doing that both in Setaria and in tomato as well. So with that, I have acknowledgments. This is my group who make it all possible. They're great, patient, and just put up with some of my crazy ideas. At Cornell, Amy is working with an Anurag's group. She works with us as well. She's very talented. This is Ganwan's group, who have been helping us with the Grape. The group at Danforth Center and in Minnesota have been involved in the CRISPR work with Setaria. And I have a collaboration with the group at the John Innes Center, and some of that funding has helped support our improvement of Setaria, because we're using Setaria as a model for that project. And these are the sources of funding for our group. And thank you, guys. Medicago takes a long time to go from start to finish. That's one of the most difficult parts. And there's one genotype that works best. So in the beginning, we had some problems because they were recommending, one of the groups was recommending using this EHA 105 super virulent agrobacterium, and we lost a lot of material from overgrowth. But when we switched to AGL1, we were able to eliminate that. So as long as you're working with that one particular genotype and you're willing to wait for quite a while, it's workable, it's doable. Not as easy as tomato, but doable. Randy?
Do you know what it is about the different gelling media that make them be good or bad?
Yeah, basically what I've seen in the reports, it's a chemical composition, but also a physical composition to it. Exactly, like Gelrite, one of the Gelang gums. It binds the divalent cations, magnesium and calcium, in order to gel. And I learned this the hard way when I was a graduate student, and I was trying some experiments with the gelling agent. I thought, why should I make up a whole batch of salt, medium with salts in it? I'll just use water and the gelling agent. And I couldn't get it to gel. I was like, why isn't it gelling? Agar gels in water. And then when I did some more searching, I found out that you do need some of the salts in there. And something like tomato does not do well on the Gelang gums because it doesn't have those calcium and magnesium ions available to it. So we end up having to use an agar-based for that. But again, it's a chemical and a physical. A lot of it has to do with concentration. We look at that also, not just the type. So. Peter?
With regard to your charcoal, I can see why you put that in to pull out some of the phenolics, but doesn't it also bind some of the hormone materials?
It does, right. So if we're working with a medium and we see that IAA works really well. I'm sorry. So Peter was wondering, you asked about the activated charcoal, that it can also bind some of the hormones in the medium, and it does. So we find that, we'll work out a system and we find out that something like IAA at a certain concentration works really well. But if we then decide, there's some browning or we want rooting, and we switch to using activated charcoal, we don't see the same response as we did with the original concentration. So then we have to bump that up and look at other concentrations. So yeah, it can have an effect. Dick?
I asked, does the monarch butterfly feed on any of the species of milkweed that you're doing? And I asked because they say monarch butterflies are declining because people are destroying milkweed in their home.
Oh, okay. Yeah, I don't know if it does on other milkweeds. I know that it uses that one primarily, but I don't know about the others. But I'm planting it in my garden now, so I'm trying to help out, and we'll see. But yeah, they're very pretty. They're very beautiful. But people think it can be a weed, but in some gardens. But
the guy across the street has wild milkweed, and it's just spewing this white stuff all over.
Oh yeah, it can be pretty messy too.
I don't mind anything for the monarch.
Yeah, that's right. Yeah, exactly. So Jin was asking about ploidy level in the plant material, and when we go through that callus and regeneration, can that have an effect in the end product? And yes, it can. And some plant species are more prone to it than others. So you'll see, I think something like pineapple, where you'll see more of a ploidy problem in regeneration and transformation than some other systems. But what some people are doing is looking at doing chromosome counts or doing some sort of an analysis to see what the ploidy level is in the resulting plants. So you have to be aware of that for some plant species because it can be an issue. Does that answer your question? Is it, yeah?
Kind of a follow-up, do you ever use pollen to get a haploid count?
We do, so I didn't talk about that, but we have done cultures to be able to get haploid material and using pollen or culturing anthers at a certain stage of where the pollen development. So we had a project with rice where we were developing haploids. And actually I was talking with a grape breeder just yesterday who asked me if we would consider doing that with grape, is looking at haploids for grape and using a pollen system, because grape is such a heterozygous material, it makes it difficult for both sequencing and for doing their work, but if they had a haploid type grape, that it would really help advance their work. So yes, definitely use that. That'll take some work to try to get that worked out.
Have you done any maize or rice?
I did maize as an undergraduate, and that was enough of an exposure to maize. Now, so actually, yeah, I'm a Setaria now. But actually, I kind of enjoyed it because we, at certain times of the year, they would pull everybody into a lab. I was an undergraduate, work study student, and for maize, the starting material are immature embryos, and you have to have the plants at a certain stage to have these immature ears. And then what you do is you cut the kernels and you go in, you scoop out the little immature embryo, and it depends on how it's oriented that you'll get the right type of callus. And when we started our transformation service, David Stern, said, well, what about doing maize? And I said, oh, that's really tricky because you basically have to dedicate greenhouse space to have the plants, to be able to get your ears, to get your immature embryos. So it kind of becomes a financially difficult, space difficult to be able to do maize. And sometimes you grow those plants and then you don't have any requests for doing the transformations, but you need to have that constant supply of ears.
So what's the difference between maize and Setaria then? If you can use Setaria just to seed, but not for maize?
Yeah, it's something genetic and physiological about maize versus Setaria. Yes.
And rice?
And rice, you can start with mature seed. So mature seed, you culture it like Setaria, and you get that, you get that embryogenic callus from it. Someone asked me, have you considered doing immature embryos from Setaria? You saw how small those seeds are, and I said, I think I'd have a mutiny on my hands in the lab if I asked people to take immature embryos out of those small seeds. But fortunately, like rice, the mature seeds for Setaria works well.
You haven't been asked to do rice?
We just did rice as part of an NSF project a number of years ago, but I haven't been asked otherwise here.
Big deal.
Yeah, so nobody has asked us to do it. So and what we did when David asked me to start up the transformation service as part of my research program, I talked with the project leaders at BTI and I said, what are you interested in? What type of crops would be important for you? And we based it on their interest first. And since that time, we've done work for people at Cornell and also outside of Cornell, but rice hasn't been one of those that we've done.
I can't remember which species it was where you used the filaments.
Yes.
Grape. Is there any particular cell that's giving rise?
I'm sure there is. I just don't know if anyone has looked at it because like in rice, they've shown, and in maize as well, it's the scutellum where the callus originates from. So I'm sure there's got to be a particular cell type in that connective tissue.
The reason I ask too is because when I want my students to look at meiotic chromosomes, but we fail because we don't have the right stage, what we can always look at are the shape of the nuclei in the filament. And they're so different. There's such a variety of shapes.
Oh, that's interesting. Yeah, so there must be something to that. That's great.
Sorry, out in left field, but in March it was announced that all cultivated sweet potatoes have the genes for agrobacterium in them.
I was thrilled.
And the wild ones don't, so there must, and they thought it's thousands of years ago that it got infected. What possible advantage could agrobacterium have?
I agree. I wondered the same. What was it through? Not the wild species, but the more cultivated. Oh, sorry. I'm sorry. I get so excited. There was a recent paper that showed. Actually, it was from the International Potato Center, where they were looking at sweet potato and doing kind of transcription profiling to find out, and they found sequences from Agrobacterium in some of the sweet potatoes. I think they tested like 300 different types of sweet potatoes. 291.
And that was every variety that they tested that was a cultivated variety,
Mm-hmm.
but not the wild one.
Mm-hmm. So there must have been some, I don't know if the cultivated ones or the wild ones had some sort of resistance to the infection. If the cultivated ones, that resistance broke down, and then they were infected. I don't know if there's an evolutionary benefit, for the Agrobacterium and the wild sweet potato and the cultivated. But I was happy because I thought, okay, what are they going to do about GMOs now? With sweet potatoes, and they have these components of Agrobacterium. How are they going to classify? How are they going to classify that? So, but it was interesting.
Incorporated into the genome, or?
Yeah, mm-hmm. Yeah, it's incorporated into the genome. Yep.
It's true for mistletoes and oaks.
All right.
If mistletoes.
Oh,
and
really?
oaks, there's at least 5 different segments of the oak host that's now integrated into the mistletoe genome.
Oh, interesting.
And it might be this kind of notion of recognizing self and immunological phenomenon.
Okay. Yeah. Yeah, it could be.
Oh, that's a
Could
question.
be.
How easy are sweet potatoes to transform then? They should be
You would
super
think it'd be easy. Just get the tuber, mix it with aggro, you're done. Oh, you get plants back from it. But we haven't worked with sweet potato, but I think certain species are easier than others. But it might be more amenable to it. Yeah. Okay. Well, thank you, everyone. Thanks.
This has been a production of Cornell University, on the web at cornell.edu.